All right. So we will be formally beginning the session now. Namaskar to everyone. Respected Vice Principal and the Head of the Microbiology Department, Dr. Shantini Nair ma'am. Former President and Trustee of IWSA, Dr. Surekha Zingde ma'am. Former President IWSA, Dr. Susan Ipien ma'am. Our speaker for today, Professor Apte, UMDAE, Center for Excellence in Basic Sciences, all the teacher and student participants who have joined here. A very good morning to all of you. It's a pleasant morning. It's at least pleasant here in Mumbai, and it's an excellent time to assimilate knowledge and gain benefits from today's session. I, Dr. Shweta Patil, on behalf of the Department of Microbiology at VES College of Arts, Science and Commerce, we are all happy to host the session today on the world's most radiation-resistant bacterium, Dinococcus radiodurans, and what you can do with it under the Popular Science Lectures organized by Indian Women Scientists Association IWSA, supported by the Board of Research in Nuclear Sciences BRNS, Department of Atomic Energy. The Department of Microbiology has been conducting many webinars under the Inspire Me Scope series and also the Popular Science Lecture series with IWSA. Through these sessions, we invite eminent scientists and distinguished alumni all over India and abroad to interact with our students and invited participants. This time also, we are privileged to host a webinar in collaboration with IWSA, that is Indian Women Scientists Association, under the Popular Science Lecture Series. May I now request our Vice Principal and Head of the Department, Dr. Shantini Nair ma'am, to deliver the welcome address. Over to you, ma'am.
Thank you, Shweta. Honorable Dr. S.K. Apte, our resource person of the webinar today, respected former presidents of IWSA, Dr. Susan Apen Madam and Dr. Surekha Zingde Madam, all our esteemed invitees, dear faculty members, and my dear students. Good morning to one and all. On behalf of our principal, Dr. Anita Kanwar, and VES management, it gives me pleasure to extend a warm welcome to each one of you. To speak very briefly about our college, VES College of Arts, Science and Commerce has recently been granted the autonomous status by UGC and University of Mumbai for a period of 10 years. The microbiology department at our college has great pride in having many of our alumni at prestigious institutes all over the globe. To name a few, we have students at Harvard Medical College, Sanford Burnham Presby's Medical Discovery Institute, California, Center for Tropical and Emerging Diseases at University of Georgia, Washington University School of Medicine, Johns Hopkins School of Medicine, Duke University Medical Center, Ludwig's Maximilian University, Germany, and many more. Our alumni stay connected with us, and in fact, we are proud to even mention here, if we may, to have one of our eminent alumnus, Dr. Subhojit Sen, who's a Ramalinga Swami at the Center for Excellence in Basic Sciences, University of Mumbai DAE body, who is a member of our BOS in microbiology along with 4 others. I express my appreciation to the convener of this webinar, Suman Gangar, to bring about this connect under the aegis of IWSA. We are indeed very delighted to have such a brilliant, distinguished scientist, Dr. S.K. Apte, whom we all know, addressing us today. We at VS College have had the privilege of learning from him on more occasions than one, and I do remember a session during our UGC-sponsored seminar in the past on building scientific temper among students and teachers of higher educational institutions. I still remember very vividly the inspiring session conducted by Apte sir then. Our college has always tried to promote research, not only at the PG and PhD level, but even among our young UG students. Our college has had the unique distinction for being awarded the overall championship in pure sciences category for not one, but 2 consecutive years from 2000 1920 and 2020-21 in the prestigious Avishkar Research Convention conducted annually by the University of Bombay. At the end, I would like to quote our former President of India, Sri Pranab Mukherjee, who said, "Science, education, research, and innovation are the four pillars on which the development as well as the work culture of a nation rests. Scientific temperament cannot happen unless we improve the delivery of education at all levels." So, dear students, with that quote, I'm confident that the webinar being delivered by our distinguished resource person, Dr. S.K. Apte today, will have a great impact on your young minds. And what you will be learning today from Sir will certainly go a long way in broadening your knowledge and thinking. Looking very forward to the session, sir. Thank you.
Thank you, Shantini ma'am, for giving the welcome address. Let me now request Dr. Surekha Zingre ma'am, Trustee, IWSA, to brief us about IWSA and a glimpse of the activities conducted by IWSA. Over to you, Surekha ma'am.
Thank you, Shweta, and thank you, Shantini and Dr. Nair. It's a pleasant morning and good morning to each one of you. I will just take a minute to bring up the presentation so that you're with me as I go ahead and talk. Is that visible now? Just confirm, please.
Yes, ma'am.
Yes, ma'am.
Yeah. So welcome to all the participants of this program. And we join hands with Vivekanand Education Society, colleges, and look forward to this wonderful lecture that is to follow, being delivered by Dr. S.K. Apte. But before we move on to that, I would like to take a few minutes from you and introduce you to the Indian Women Scientists Association. On behalf of the trustees, I'm one of the representatives from them, the members from the executive committee, and the IWSA members from headquarters and the branches. Hello, can you see that or it's gone?
Yeah, ma'am, slide is not visible now. Hello?
Yeah.
Yeah, ma'am, can you reshare it maybe?
Yeah, can you see that?
Yes, we can see, ma'am.
All right. So to move ahead, IWSA is an NGO, non-political, secular, and social welfare organization. We are based out in Vashi, which is our headquarters. You can see it out here. And we are moving into our 50th year. As we celebrate, we bring out this logo, a time to look back, to move forward, and to move ahead. We are registered with the Niti Aayog as is per requirement of the Niti Aayog mandates. IWSA is in its 49th year. We will be having our 50th year in June of 2023. That is when we complete 50 years. During these last 50 years, we have really moved ahead, and we look forward to moving and soaring ahead in the years to come. With the headquarters in Vashi, we have 11 other branches, which you can see on the right-hand corner, right from Roorkee in the north to Nellore in the south. About 2,100 IWSA members. Main mandates is to take science to society and for women empowerment. Our two arms are community welfare and science awareness. And during 2019-20, we reached out to about 23,000 recipients. When we mention recipients, we mean individuals like yourself who were beneficiaries of scientific programs that we brought out with them and to them. And even during the pandemic times of 2021, we have not done very badly. We were at 1,700 numbers. Among our community welfare activities, we have a working women's hostel, a healthcare center, computer center, a library, a program which is referred to early childhood care and education, and a scholarship awards program. For those young girls who are in the audience, we have this working women's hostel started way back in 1989, and we can house up to 160 working women. We follow the guidelines of women and child welfare, and it is a clean, economical place. So do encourage your friends who are looking for a place to stay. Through healthcare, we'll take out cancer camps and other clinical investigations. And at the center itself, we have a dentist and a physiotherapist. A computer center to encourage youngsters as well as senior citizens to learn the use of the computer and all the new apps that are coming about. A library supported by the state government. A scholarship awards we just completed this year. This is run through donations received from well-wishers, and from the interest we support and provide awards to youngsters who have completed their 10th standard, 12th standard, BSc, MSc and PhD. And we just had this last week with Dr. Vasudha Kamath being our chief guest, and you can see them being receiving their awards. The other program is the ECC program, which I will speak to you a little later. And a daycare and nursery school, unfortunately, at this point, during the pandemic, it is temporarily closed. The computer center has completed 30 years, and we had a lovely program on changing face of digital era, looking back and looking forward to see what we could do through the computer center. The early childhood care and education program is a one-year diploma in affiliation with the SNDT Women's University. And any girl who is 12th standard pass is eligible to join. Age is no bar, and they would get this diploma where they can then go and teach in daycare centers or even pre-primary schools. And in addition to that, we offer a program along with Drishti and other NGO to train for shadow teacher training and also to understand learning deficiencies. So these programs are vocational programs as per the requirements of the new education policy. So do encourage your friends also to consider them. Our academic year starts in June, and we are look forward to having many youngsters join us. Age is no bar, as I said, several times senior ladies also join this program. This program also has received attention from Vigyan Prasar, and we have conducted a 30-hour course, Shikshan Setu for pre-primary and primary teachers, which is online, and 6 modules of the course content are prepared by our own ECC teachers. So the teachers benefited a lot. They understood how teaching has to be done for these little children in early years. Science education initiatives is our next alternative. We have school students whom we address. We have a little science laboratory. Here you can see our volunteers showing them experiments. We have a science nurture initiative where we encourage youngsters from neighboring schools who need a little helping hand to do better at their whatever they are taught. We have a wonderful band of about 13 volunteers who teaches them and goes over the syllabus that they have. And the youngsters come out very confident through these interactions. We conduct science lectures for them, science camps and clubs. Some of these programs which are now under receiving support from Vigyan Prasar are indicated by VP. And of course, we have science exhibitions during the time of the Science Day. We are reaching out to college students through this program, the Popular Science program under BRNS. And we also reach out to through lecture workshops and refresher courses for faculty and for students through the Science Academy support. And during the pandemic time, we didn't stop. We had some of the youngsters come to us with online teaching. We have a learning garden, as we mentioned, and we have about 500 to 600 different plant species. And when the times are better, we look forward to welcoming you on campus to show you around. We have visitors. We take this information out to exhibitions also. And we have been able to do very much more, which I will show you in our next slide. And under the green initiatives, we have turned our campus into, through the solar system using the electricity that is generated and the solar water heater. So projects in collaboration, Vigyan Prasar considers each one teach one, where a college student and facilitates the learning process for a young school student. And here you can see that VS College also participated in this program along with Somaya College to teach some school children in the neighborhood. And another one is right now ongoing with Sophia and Mitibai colleges. And through the Vigyan Prasar, we have BIPNet, which is a science club wherein young students are shown a variety of experiments on a monthly basis. And we conduct garden-based workshops on a variety of topics which are of vocational interest to several people who would like to undertake these garden-based activities on their own or to start up little entrepreneurships on their own. We have been doing internship programs with college students. The first one was actually started way back in 2020 or so. And with that was a college, Khalsa College, where some of the interns have actually done a MSc thesis. And then IWSA, SciScom, Flame University, Jai Hind College, and now SIS College is on this program with us. And in fact, in the next 2 days, they are going to complete that program and have a complete internship with us here. During this time, we have expert lectures online, and whatever they put together is reviewed by experts from India and abroad. And some of them, through Jai Hind College, in fact, have brought out booklets of the work that they did. So it's been a very exciting time, both for the college students as well as for the IWSA members under whose mentorship they conducted different projects. BRNS supported lectures. This has been an extremely long interaction with BRNS. We started this about 11 years ago. And we have been reaching out to colleges during this time. And during 2021, we reached out to nearly close to 6,600 odd lectures, individuals through the headquarters and the branches, over 60 lectures, anything in physics, chemistry, biology, environment, and engineering, and speakers from academic institutes of repute. And today here we are with Dr. Apte. This is our 20th lecture. We started in September 21 in this financial year, and we have many more already lined up, starting one after another from tomorrow itself. And we are now under this program of 21-24, we are reaching out to schools and to society. So such lectures are being conducted in the schools. And the last lecture was with Dr. Mukesh Roy on fun with physics at St. Mary's Multipurpose High School. And in Science for Society, we reach out to all the lay people in the society, anybody interested in different topics in histories, health, education, whatever be. Here, this talk on lifestyles and aging was given by Dr. Krishnamurthy, and we also had a lecture on Dholavira by Dr. Nigam the month before that. We have only very briefly told you about what we do. All our details are available in our newsletter, which you can download from our website. And information about our branches is also there. They do in parallel many of the activities that we have at the headquarters. And those of you who are our members, if you do not receive these the newsletter, do inform us, or you can just go to website and download it. And these have been continuing during the pandemic times. So IWSA's efforts are towards knowledge sharing, bringing about a change, leading to value creation and promoting innovation, with emphasis on science-based methods. And we request you, all the graduate students in this audience, that girl graduate students, to join us as a member. Our programs are supported through donations and government grants for specific projects. But you could also help us by joining hands in our activities. Our contact is here. You can take a picture of this of this particular page, or connect to IWSA.net for more information, and help us to move forward as we look to soaring higher and higher during the next 50 years that may follow. We are already in the middle of this new year, but I still wish all of you the very best for the year to follow, and hope all of you do very well. And I'd like to take this opportunity again to thank Dr. Apte for agreeing to deliver this lecture, and to VS authorities for accepting to host it. Thank you so much.
Thank you, ma'am. We are extremely privileged to be collaborating with IWSA. IWSA is associated with all generations, toddlers to senior citizens, not a single area where IWSA is not working. Many of our students have benefited from various scholarships, workshops, and seminars conducted by IWSA in the past, and we were also associated with your EOTO activity this year. We look forward to many more interactions and always will be connected to IWSA. Now a glimpse of today's session, which we are so longing waiting for. The world's most radiation-resistant bacterium, Dinococcus radiodurans, and what you can do with it. I'm sure that most of us have heard about this bacterium, but Sir will unfold many mysteries during today's session. As quoted by Mary Engelbrett, if you don't like something, change it. If you can't change it, change the way you think about it. And I would add to this, adapt to the change. Today's bacterium under discussion, Dinococcus radiodurans, is an extremophilic bacterium and one of the most radiation-resistant organisms known. It can survive cold, dehydration, vacuum, acidic conditions, and therefore is also a polyextremophilic organism. There is a lot to learn about the adaptation mechanisms of this organism, and it has also been listed in the world's toughest known bacterium in the Guinness Book of World Records. A small announcement before we begin to the participants. Please post your questions in the chat box. Our moderators will pick the questions in the end. Without much ado, let me now call upon Dr. Susan Epian, former President IWSA, to introduce the speaker for the session and also request Sir to start the session. Over to you, Susan, ma'am.
Thank you, Dr. Shweta Pati. Good morning, everybody. It gives me great pleasure to introduce my former colleague, Professor Srikumar Apte, currently Distinguished Professor at University of Mumbai, Department of Atomic Energy, Center for Excellence in Basic Sciences. He did his MSc in Botany in Jivaji University, Gwalior in the year 1972, and he was a gold medalist in the science faculty. Later, he did his PhD from Gujarat University, Ahmedabad in 1985. Meanwhile, he did his training school course for 1 year in biology and radiobiology and passed out in the year 1973 and then joined Baba Atomic Research Center as a scientific officer. Ever since, he has served in different capacities for 42 years. He is superannuated as a distinguished scientist and director, Bioscience Group, and head Molecular Biology Division, BRC, until 2014. He was Emeritus Professor of Homi Bhabha National Institute from 2014 to 19, DST JC Bose National Fellow from 2009 to 20, and Raja Ramana Fellow of DAE from 2015 to 2018. He is a fellow of various science academies like Indian National Science Academy, New Delhi, Indian Academy of Sciences, Bengaluru, National Academy of Sciences, Allahabad, National Academy of Agricultural Sciences, India, and Maharashtra Academy of Sciences. He is a recipient of Professor J.V. Butt Eureka Forbes Award for Excellence in Microbiology, Young Scientist Award, Cyanobacterial Biotechnology, and Professor K.S. Bilgrami Memorial Award of INSA and also from Indian National Society Award in 2006. He is a recipient of several fellowships like IAEA Fellowship, then a Newfield Foundation Fellowship of UK, USAID Fellowship of USA, etc., etc. He represented India at the United Nations Scientific Committee on Effects of Atomic Radiations in 2014. Dr. Apte coordinated a DAE-sponsored multi-institutional project entitled Thermal Ecological Studies. He was a member of several selection and evaluation committees of INSA, TST, DBT, CSIR, ICAR, and DAE, besides 6 research institutes in India. His research interests vary from response of organisms to ionizing radiations and other environmental stresses to development of eco-friendly biotechnologies for agricultural applications and for bioremediation of nuclear waste. Currently, Dr. Apte is engaged in popularization of science and enhancing public awareness about nuclear energy in India. Today, Dr. Srikumar Apte will speak on Dinocockers radiodurans, the world's most radio-resistant bacteria. Dr. Srikumar Apte, please.
Thank you, Dr. Epan, and good morning, everybody. I'm thankful to Indian Women Scientists Association for inviting me to give this talk. In fact, I gave my previous talk in IWSA forum more than 10 years ago when I visited their Vashi campus and got to see the whole range of activities that they were doing at that time, and very happy to learn from Dr. Jingde's presentation that they have expanded tremendously. Congratulations to IWSA, and it's also nice to be invited a second time. I also am thankful to Vivekanand Education Society for hosting this event, for making very nice arrangements, and to everybody who spoke before me and said very nice things about me, which reminds me that I am older than I feel, and raising some expectations about what I will talk. I hope I am able to do justice to them. I will now open my presentation, and I request to everyone to please switch off their videos and mute themselves, because the number of participants seems to be large and we don't want to burden internet too much. I'll switch off my video as well. Is my presentation visible? Can somebody please confirm?
Yes, sir.
Okay.
Yes, yes.
Yes, it's
Thank
visible.
you. Thank you. So today I'm going to speak about world's most radiation-resistant microbe, as Guinness Book of World Records puts it. The name is Dinocockers radiodurans, and the organism is by now almost well known. I can take a small credit for bringing this organism and its related research to India, and now lots of labs are working on it. Why it is so radiation resistant is the area on which most efforts have been spent so far, but I have talked about that many times. So today I will spend a little more time in telling you what can you do with such a wonderful organism. This slide also gives my email address, so that some of you, if you are not able to ask your questions today, feel free to write to me on my email address. So let's get on to Dinocockers radiodurans straight away. This is how the organism looks. Its colonies look on a plate, on a Petri plate. And this plate has a date which tells you the first time I ever plated out this organism way back in 2003. Very nice, deep orange or red colored colonies. That morphology is what gives its half of its name. Dinococcus in Latin means strange red berries, and that's what it looked like to microbiologist. It was isolated first time in 1950s from irradiated meat cans, and later on in 70s in BRC as well from irradiated meat products by Dr. Lewis. Its genome was sequenced somewhat later in 1999, and since that time there has been a spurt on research in these organisms in various directions. In a scanning electron micrograph, it presents some very interesting morphology. These grows as tetrads, 4 cells together with very interesting surface structures on them. We still don't know what they are. But the most famous thing and most impressive thing about its lifestyle is its extreme radio resistance, which is shown on this slide at the bottom on the left-hand side, which compares survival of different organisms with increasing dose of ionizing radiation. As you would expect, humans are the most sensitive. The first line, which is almost on the y-axis, being the most sensitive to ionizing radiations, followed by the world's most hated animal, cockroach. I'm sure all of us want to kill it, but as you can see, even by radiation, it's going to outlive us because it's doing better than humans. Bacteria are somewhat more resistant than other organisms. And over here, the red line represents one of the most sensitive bacterium, E. coli, which tolerates less than 1 kilogram of radiation. Plants do little better. Plants and insects do little better than this, but nothing compares with Dinocockers radiations, whose survival curve is shown here. And up to a dose of about 6 or 7 kilogram, this organism behaves as if it has not seen any radiation and responds with 100% survival. Incidentally, this dose, for those who are not aware of what kilogram dose means, is a dose that will be expected to be found within 100-meter radius of a Hiroshima-sized bomb. Such level of radiation has never existed on our planet, incidentally, but this organism shows such resistance. And it's only at much higher doses of 15 or 20 kilogram that it starts showing some sensitivity. So because of its association with radiation, high radiation environments, irradiated meat, etc., the first thought when people discovered it was to do 2 kinds of things with this organism. One is, of course, a very novel, interesting basic research on aspects of its radio resistance, which is sort of the ultimate in this organism. DNA repair, because at that level of radiation, DNA should go to literally pieces. And oxidative stress tolerance, because radiation generates hell of a lot of reactive oxygen species. Simultaneously, people also thought of developing biotechnologies for high radiation environments where this organism has a distinct advantage over others. And so far, as I said, most of the work has concentrated on this aspect and on this, but today I will share with you some non-nuclear applications about this organism as well. Going to the next slide, as somebody mentioned, I think Dr. Nair mentioned that Guinness Book calls it the world's toughest bacterium. There have been papers in Nature and Science asking, does this organism wear a lead shield or what? Lead being the element which blocks radiation and is used often as a shield. The answer is no. If you irradiate this organism at such high doses, over here about 17.5 kilogram or more, it's literally, this DNA literally goes to pieces. This is an optical image of the DNA fragments you see at that dose, and you won't expect any such cell containing such fragments of DNA to survive. But you take out these cells, give them a wash, put it in a fresh medium, and just wait for a certain time, which depends on the length of exposure. At this dose, it takes about 24 hours. At 6 kilogram, where it has 100% survival, it takes just 4 hours to recover back. And what it does during that time is to put back each of these pieces correctly in its original position, in correct orientation, to stitch back completely intact chromosome. Now, this ability no other organism in the world has, and that's what is most remarkable feature about Dinococcus radiodurans. It has also been isolated from high radiation environments, including places where nuclear stockpiles were stored during Cold War and leaked, making the area radioactive, and Dinococcus was found even from such environments. And with these in mind, Department of Energy USA sponsored its genome sequence somewhere around 99, and that revealed some very interesting features about its genome sequence. I'm going to touch upon some of the interesting aspects only briefly, and I'll just show you some glimpses of those highlights here. So over here is the information on its genome sequence, which, as I said, came out in Science in 1999. The organism has 4 pieces of DNA, one large chromosome, one smaller chromosome, one megaplasmid, and one small plasmid. The small plasmid being the size of what bacteriophage lambda has, around 45 kilobases. The genome is smaller than E. coli. It's only about 3.28 megabases compared to 4.7 of E. coli. And at any given time, each of these pieces of DNA occurs in the cell in 4 to 10 copies. So at any given time, cell may have 10 to 40 pieces of DNA comprising each of these 4 molecules. As is typical of bacteria, 90% of this genome has genes, coding regions, but it also has a relatively high content of repeats for bacteria. So total about 3,000 odd genes. At the time when it was sequenced, nearly 2/3 of these genes were known in other bacteria as well, and they could be identified. But around 1,000 genes were totally unique to this organism with no database match in any other organism, and this number still remains more or less same, more than 950. One still doesn't know what those genes do, although a few of them have now been worked upon and identified as to their function, etc. And most of the research in subsequent years has been to discover the mechanisms underlying its radioresistance and repair, either to see if the known genes of other bacteria are responsible for such unique abilities of these organisms, or these unknown genes not found in any organism hold the key for that kind of remarkable property. Now, strangely, for an organism which is so efficient in DNA repair, it lacks some very important and common DNA repair genes of other bacteria. For example, REC-ABC pathway, it lacks REC-BC. SBC-BC pathway, it lacks SBCB, and many other genes it lacks, which is somewhat unexpected for an organism which is so phenomenally DNA repair proficient. One would expect that it would have everything that other bacteria have, plus maybe much more. And that raised an interesting question as to why such genes were absent from this organism. They are very common elsewhere. Why should they be? So one of us in BRC asked a crazy question. Is there any advantage in losing these genes? One of the such genes was, for example, SBCB, which makes for an exonuclease called exo1. It's an important gene in DNA repair. Most bacteria have it, but it's missing in this organism. So what we did was to take this gene from E. coli, tag it to a dinococcal promoter so that it can express in Dinococcus, and expressed it in Dinococcus to see what happens. And what happened was a big surprise. Compared to the normal radiation resistance of Dinococcus with increasing doses of radiation, the Dinococcus which was now given this gene from E. coli suddenly became radiation sensitive, indicating that there was some merit in losing this gene. It contributed to its radio resistance. Loss of this gene actually gained a function for Dinococcus, making it more radio resistant. The details as to why this was the case was investigated, and I am not, it's a full paper there in case you want to see, but I will just share one result with you. Of course, this did not affect the UV resistance, but then what we found was that if you irradiate Dinococcus before irradiation, that's the size of the genome on a pulse field gel, and post irradiation, after DNA has fragmented quite a bit, it goes down, and during recovery it stitches back these chromosomes, and at different points of time you can see this DNA grow in size, ultimately becoming as large as the original DNA, thereby completely restituting the genome. That's the case in wild type. But in Dinococcus, where it transferred SBCB gene, what happened was shocking. The DNA fragments, which occurred as soon after radiation, remained where they were. There was no stitching back of this chromosome and no regaining back the normal chromosome. Eventually, such cells died. And that's the reason for this radiosensitivity. And this we worked out to show that this is an exonuclease which chewed up the fragments, especially the overhangs, DNA overhangs, and didn't allow this organism to stitch back its chromosome because loss of considerable amount of DNA, and that caused its sensitivity. Now, similar studies have been done for a few other missing genes, and everywhere the result is same. There is reason why it has specifically lost certain DNA repair genes. It has gained thereby in its radio resistance. What about the other DNA repair genes? Over here is one of the early results, which was very interesting. REC-A, Pol-A, UVRA, as all of you microbiology students know, are very important DNA replication and repair genes in bacteria. Now, when these genes were individually mutated in Dinococcus, you can see that such mutants became terribly radiosensitive compared to wild-type Dinococcus, which was fairly radio-resistant, suggesting as if each and every one of these genes itself was responsible for such radioresistance, and loss of even one such gene resulted in radiosensitivity as much as E. coli is radiosensitive. Surprisingly, however, when Dinococcus genes, REC-A or Pol-A, were transferred to another organism such as E. coli, it did not increase its radio resistance in any way at all. So there was nothing special about these genes doing the same job of proficient DNA repair in another organism. They worked only in Dinococcus. Similarly, in such mutants of Dinococcus, if corresponding genes were transferred from E. coli, they allowed it normal growth, but they did not contribute to its radio resistance. So overall, the conclusion was that yes, individually these DNA repair and replication genes are important for Dinococcus, but their contribution is context specific. They need the whole intracellular environment of Dinococcus, other genes, other proteins, other metabolites that are present in Dinococcus to be able to contribute to its phenomenal radio resistance. Individually, these genes, either in Dinococcus or outside Dinococcus, do not make much sense. So that was the case with genome. What about its proteome? The radiation also affects all kinds of biomolecules, including its proteome. So this is one of the early results. This is one of the results where we compared the proteome of Dinococcus cells before irradiation with the one after irradiation. And you will notice that largely speaking, practically everything here is present here to the eye with this resolution. That appears to be the case. But you see several arrows and names here, which indicates some of the genes that were reduced after radiation and some of the genes whose expression increased after radiation. You can see it very well in well-resolved regions, but not in this region. Now, this was surprising that some of those genes were synthesized post-irradiation, in spite of the fact that DNA had undergone such extensive damage. So what we did was, following irradiation, we followed the proteome for next few hours to find that yes, some of these proteins had received damage in the form of protein carbonylation, sulfonylation, nitrosylation, etc. And they were degraded but resynthesized within 4 to 6 hours to sort of get back the proteome as efficiently as the genome, and that's why Dinococcus could start regrowing within 6 hours. So if you look at that detail over here, for example, single DNA repair genes, each of these are DNA repair genes whose protein profiles have been monitored in the early hours of post-irradiation recovery between 0 to 6 hours. And as you can see, each of these DNA repair genes starts very low, but in the next few hours, 1 or 2 or 4 hours, it increases tremendously and then comes down. And by 4 hours to 6 hours, the recovery repair of both the DNA and proteome is complete and Dinococcus is back to normal. So how under such conditions of extensive damage, this organism is able to induce expression of certain proteins and genes was a big surprise. And towards that, some answers were found later, and I'll just share a couple of those results with you. The first answer was found a little earlier, in 2003 itself, by the Chinese group of Hua. What they did was to create random mutations in Dinococcus. Incidentally, Dinococcus cannot be mutated using ionizing radiations, but it can be mutated using chemical mutagenesis. So using chemical mutagenesis, Hua created random mutations, and one of those mutations he found became radiosensitive, And when we characterized it, mutation was found to be in a gene which he called PPRI. The PPRI gene did some interesting things. So over here is a comparison of protein expression in wild type and PPRI mutant. So first and second lanes are wild type and third and fourth lanes are PPRI mutant. And what it shows that important genes like RECA, PPRA, etc., which are important DNA repair genes whose synthesis is switched on after irradiation. In wild type, as you can see, compared to the earlier level, after gamma radiation, their levels increase, but not in the PPRI mutant. PPRI mutants fail to increase the levels of these proteins or genes, which are very much required for repair of DNA, and as a result, it cannot tolerate radiation and becomes radiosensitive. That was the case also when the PPRI gene was restored back, it showed some normal induction post-irradiation. So this gene was named by Hua's group as pleiotropic protein response inducer, because it sort of switched on the expression of several genes and proteins following irradiation, and in the absence of this gene or deficiency of this gene, cells, Dinocockers cells, failed to respond to radiation by increased synthesis of these repair proteins and became radiosensitive. However, the mechanism by which PPRI did that was not known for next 10 years, and I'll share what happened after that a little later. So this was the first clue as to how the post-irradiation induction of synthesis of important DNA repairs takes place, thanks to this particular gene, which has been called a molecular switch. The second important result came somewhere around 2007, by which time 3 different Dinococcus strains genomes have been sequenced. All of them were radiation resistant. And what this group was found by large-scale genome comparisons, that upstream of close to 100, 150 of genes involved in radiation resistance, DNA repair, etc., in all 3 organisms, this particular motif was noticed. So you have a palindromic sequence. As you can see, the bases here and here are complementary. In the center of the symmetry is somewhere here. The sequence is not very highly conserved. Only the tall bases are highly conserved. The smaller bases are less conserved. But nevertheless, it makes a nice palindrome, about 18 base pair palindrome. And the interesting thing was that it was found upstream of all 100 odd genes, many of them involved in DNA repair. And they thought that this is what was responsible for post-radiation response of these genes, or for radiation induction of these genes. And similar thing was also found for desiccation, which also causes extensive DNA damage. And they called this particular motif as a radiation desiccation response motif, or RDRM for short. And strangely, this was found, as I said, in all 3 Dinococcus strains, all of which are radio resistant, and it's found upstream of hundreds of DNA repair and radio resistance related genes in all 3 organisms, earning it the name radiation desiccation response motif, and it's a 18 base pair palindrome, as I said. Now, the first evidence that this indeed serves that function was obtained in our laboratory by this particular paper, Aman Ozaune, who found that this particular motif, indeed, if you mutate it, the genes lost their radiation response. He showed it for the SSB gene, single-stranded DNA binding gene, and later on we found it to be the case for many other genes as well. So what we did, by that time we started an analysis of several dinococcal radiation-induced promoters. And what you see here is a result where what we did was we took all these promoters. These are the names of the various DNA repair genes that you see here. And what we did was to take promoters of these genes and tag them to a reporter gene like green fluorescent protein, so that whenever the promoter expresses, the green fluorescent protein will express. And the advantage of GFP was that we could visualize it directly in the cells, as well as quantitate the activity by measuring GFP fluorescence. And one thing we noticed was that in all the genes which contained RDRM, the basal level activity of the promoter was rather low. You saw hardly any GFP fluorescence in these cases except this. But after we irradiated them, each and every one of them showed several fold higher fluorescence, and all of them fluoresced bright green compared to without radiation. This suggested as if the RDRM was normally keeping the expression of these promoters low, but post irradiation somehow that repression was alleviated and GFP started expressing because the promoter got activated. How much were these promoters activated by radiation? Well, we quantitated them and they ranged from anything from 4 or 5 fold to up to 25 fold. In fact, now we have promoters which are induced even 40 fold. So all these promoters are normally expressing at low level, but post irradiation, they are expressed at very high levels. And these are the genes whose synthesis goes up in the early hours of post radiation recovery and restore the levels of these DNA repair proteins, making Dinococcus capable of post irradiation recovery. Meanwhile, two groups, one in France, other one in China, found some other interesting results. One of the groups was working on a protein called Ddro because it looked like a repressor. It resembled DNA expression repressors in many organisms, and they thought they should look at it in more detail. What they found was that in vitro, with purified DNA, Ddro bound the RDRM regions, suggesting that it may bind there and repress the expression of the corresponding genes. Immediately thereafter, Hua's group, Wang et al., tested the PPRI, which they had identified earlier as the molecular switch, to see what it does to this Ddro, and found that in vitro, if you add PPRI to DNA bound to Ddro, it cleaved Ddro, suggesting the possibility that normally why these genes are expressed at low level is because Ddro is bound to them. But post irradiation, PPRI gets activated, acts as a protease, cleaves this Ddro, and the expression of these genes start because RNA polymerase can now bind there and do this. Now, all this was suggested in 2014 and 15 in vitro. Now, it was another task to prove that this model works in vivo. But since by that time we already had several RDRM containing promoters, we tested it in vivo and proved this model in vivo. I'll just show you 2 slides of that. So what we did was to take this whole system to E. coli. The reason being that E. coli is a clean system. It neither has Ddro nor has PPRI. So we can start on a clean set and test all these hypotheses. So into E. coli, we moved our various promoters tagged to GFP and found that they expressed in E. coli also, some of them. Some of them expressed very highly, others expressed low. Look at the blue bars here. And then into this E. coli, we express Ddro from Dinococcus. So these are E. coli expressing dinococcal Ddro, the so-called repressor proteins. And you see that in every single case, after Ddro expression, the expression of fluorescence came down significantly, clearly showing, therefore, that Ddro of Dinococcus is able to repress activity of these Dinococcus promoters containing RDRM even in E. coli. So yes, Ddro can function as a repressor of these genes in vivo. So second thing was to prove that PPRI can also work in vivo, for which we created a PPRI knockout mutant of Dinococcus radiaturans. So we created a Dinococcus which had PPRI wild type and one which lacked PPRI. Now into these strains, when we moved some of our promoters containing GFP, they showed their normal fluorescence in the absence of radiation, indicated here by the red bar. But after irradiation, that level remained same. There was no induction in GFP expression after irradiation. And why? Because that was brought about by PPRI. And since we had knocked off PPRI, there was no radiation-induced gene expression in these organisms. Now, that sort of explains 3 unique things about it. First is that Dinococcus is one unique organism where there is clearly established radiation-induced gene expression, which has been suspected in many organisms, including human cells, but never very clearly demonstrated. Here it is so nicely demonstrated. Secondly, this operates through the Ddro PPRI system. And thirdly, that is what is responsible for suddenly switching on expression of many of the required DNA repair oxidative stress alleviation genes after radiation, restores their levels, and Dinococcus is able to repair its genome and starts its life all over again. That's the essential story behind why Dinococcus is so radio resistant. There are many other aspects about it, but I would now rather tell you more about what can you do with such a wonderful organism. So first and foremost, of course, because of its association with radiations, one of the first things that occurred to people was to see if they can engineer this organism to do things for high radiation environments, including for nuclear waste. Nuclear waste, as all of you know, contains all kinds of undesirable things: uranium, decay products of uranium, lots of organic solvents, and so forth. So within 1 year of the genome sequencing of this organism came the first paper in Nature, which I am not showing you, which showed that Dinococcus could be engineered using mercury reductase genes and toluene degradation genes from E. coli and Pseudomonas, and removed these two toxic elements from nuclear waste. So we thought we could use it for removing uranium from nuclear waste, and the next few slides will show you that success story, which worked very well in our hands, and I just will show you some of that. The idea behind this was to use a phosphatase, shown here. In the periplasm of bacteria is usually present a phosphatase, which if you give it an organic phosphate will cleave it to generate inorganic phosphate. And what's the interest in phosphate? Is because uranyl phosphate makes a very, very insoluble permanent precipitate. So if you can precipitate uranium as uranium phosphate, it doesn't redissolve and get back into water. So if you can precipitate uranium as uranium phosphate, you have essentially removed it from aqueous waste. And the radioactive waste that one generates is usually of two kinds: one acidic waste containing uranyl nitrate, one alkaline waste containing uranyl carbonate. So we were looking for an acid phosphatase as well as an alkaline phosphatase, which will do that job in the respective pH. So one such acid phosphatase was 4N, another one alkaline phosphatase was 4K. So we engineered first E. coli and then Dinococcus to express these genes in them and look at what they will do. First, of course, was tested with E. coli, but I'm showing you the final result with Dinococcus. So how you do it, of course, is you transform Dinococcus, you streak it on plates. Normally the color is orange, as I said earlier. But then now if you convert this plate into what we call a histochemical plate, where you put methyl green. Methyl green is a colorless dye, which is an indicator of acidic pH. So when the phosphatase works and produces phosphoric acid, the pH around the colonies goes acidic, which is picked up by methyl green, which suddenly changes it to green color, indicating that yes, these colonies are now producing phosphatase and producing and generating phosphoric acid. So that's the first initial test that your construct has worked. And now look at the ultimate result. We tried to remove uranium from aqueous waste, in this case at acidic pH, at concentrations way below 1 millimolar. Why? Please remember here the idea is not to concentrate uranium into kilogram and ton quantities to use as fuel. Idea is to remove traces of it in sub-millimolar concentrations from radioactive waste so that the waste can be disposed of easily. And at that concentrations, physicochemical methods don't work. That's why this enzymatic approach was used. And when we did that, what we found is within few hours of time, both E. coli, the orange line, or Dinococcus, yellow line, which were engineered to express phosphatase, acid phosphatase, could remove more than 90% of uranium at that sub-millimolar concentration within few hours of time. But we wanted this to work in radioactive waste. So you now applied radiation. Dinococcus still continued to do it, albeit at a little less efficiency, but E. coli simply perished at that level of radiation. E. coli cells lysed, completely fragmented, the phosphatase was lost, and such E. coli cells couldn't do anything. So that sort of provides the proof of principle. While the method works in E. coli as well, you can use it for non-nuclear situations. For high radiation environments, necessarily you have to go to a radio-resistant organism like Dinococcus. Where was all this uranium going? So over here are lyophilized Dinococcus cells, tetrads, as you can see. And what we are showing you here is an SEM picture showing X-ray diffraction analysis of that to show you what kind of elements it has. And over here is Dinococcus lyophilized cells after they have been treated with uranium and have removed uranium. And they look like this, a fluffy stuff sticking all around cells. And we hoped it was uranium, and it was indeed uranium, as this spectrum showed. There are several uranium peaks noticed here. So it indeed was uranium, and this presented very interesting morphology. One of the seminars which I was giving to 10 standard students in Mumbai, the student told me it looks like sev batata puri, which is a very common dish in restaurants in Mumbai, for example. I don't know if it looks like that or not, but it certainly looks very interesting, and all of it is loaded with uranium. Interesting thing is uranium binds to this membrane, and then the precipitate grows outward. So normally Dinococcus is sensitive to uranium, but because all this uranium gets sequestered outside the cell and is kept outside the cell, such cells can continue to grow in uranium and precipitate uranium. The other important thing is that if you now take a transmission electron microscope picture of that, you see this needle-like crystals of uranium sticking all around the cells. Very little in the free solution, most of it sticking to the cells. And this offered a tremendous advantage. Since the precipitated uranium was bound to cells, cells became heavy. Uranium is a very heavy metal. Cells settled down, and within 2-3 hours of time, the entire cell mass along with uranium settled at the bottom of the waste. You can then decant the supernatant, collect the cells, and either dispose them or recover uranium from them. So it became a very easy technology to operate. Otherwise, you have to centrifuge huge volumes of liquid waste to isolate the precipitate from that. Now, what about alkaline conditions? Now, in alkaline conditions, uranium carbonate is very highly soluble. It's almost impossible to precipitate that unless you generate 1,000-fold excess of phosphate, and for which we found a very novel enzyme from a bacterium called Novosphingobia. This by far is the highest specific activity bacterial phosphatase. We cloned that in Dinococcus, and that could also precipitate uranium under alkaline conditions. Now, when we tried to publish this paper, which eventually got published, one of our reviewers was a hardcore chemist, and he said, while I am impressed with your result, I don't believe that what you have precipitated is uranium, because unless you provide 1,000-fold excess of phosphate, uranium will not precipitate. So please show me doing XRD that it is indeed uranyl phosphate. So that forced poor biologists like us to apply biophysical methods. We did XRD and got this spectrum, which promptly tallied in the database with this particular form of uranyl phosphate. It has 8 water molecules of hydration, and this form of uranyl phosphate is known in uranium literature as a meta-autunite or Chernikovite. Needless to say, the paper got accepted, but it also provides us a very interesting information. Meta-autunite or Chernikovite is a form of uranyl phosphate on which if you shine UV, it fluoresces green. So we said, wow, that's a very nice handle to detect if uranium has been precipitated or not. And the PhD student who worked on this, incidentally, was a Bombay University PhD student with me, Kaizad Nilgiriwala. And you can see here that compared to the normal Novosphingobia from which we isolated this enzyme to the engineered strain which express this enzyme very highly, you see a green precipitate after uranium is precipitated, but not in other kinds of controls. You can see it from top, you can see it from sideways, whatever. And this is fairly intense. For example, if you precipitate uranium from 1 liter solution and leave it in a room, it will easily light up at 15 by 15 lab in the night, almost like a bright night lamp. The interesting thing about this precipitate, however, is that it doesn't bind to the cells. It is found in solution. And that's because the alkaline phosphatase, which we use for this purpose, sends out, is released extracellularly. So it precipitates it in the extracellular medium. And that creates one problem, how to now trap this particular precipitate, which was easy in the case of acid phosphatase because it was cell bound. So here what we did was to immobilize Dinococcus cells expressing alkaline phosphatase into calcium alginate beads like these. So these are beads. These are beads containing Dinococcus expressing 4K. And then these are the same beads after they have precipitated uranium. Shine UV on them and you find all the beads fluorescing bright green because of the uranyl phosphate metaautonite precipitated in them. And this shows you the time course of it. This reaction happens very fast. Within about a couple of hours time, you remove almost more than 90-95% uranium from it. You can measure it using chemical methods. You can measure it using fluorescence or whatever. So this method works very, very well. Now, whenever you give extra genes to an organism, it sometimes compromises its existing ability. So one of the questions one had in mind was, by giving such extra genes, phosphatase, alkaline or acidic, which one came from Salmonella, other came from Novus pingobium, and the cell has to now overproduce this, will it burden the organism's existing capabilities, in particular its radio resistance, because we didn't want the radiation resistance compromised. After all, this had to work in radioactive waste. And over here is an analysis to show that with increasing doses of radiation, the survival of Dinococcus expressing 4K remained same as that of the wild type, and its ability to precipitate uranium also remained same with increasing concentrations of uranium. Therefore, the addition of these genes did not compromise the existing radio resistance, and even irradiation did not influence bioprecipitation. Now let me turn my attention to some of the non-nuclear applications. It's not that because it's radio resistant and therefore very relevant to high radiation environments, it cannot do other things. It has some very other interesting molecules, and I'll spend next 10-15 minutes before I close on such molecules. One such molecule is the orange pigment in Dinococcus cells, which provide its given color. It's a xanthophyll, which has been named dinoxanthine. Dinoxanthine has been found to be a very potent xanthophyll, almost as good as lycopene that we get from tomato. Incidentally, Dinococcus also makes lycopene. And these kind of carotenoids act as excellent raw scavengers. And the molecule, the pigment, has been tried many times in many situations and show that they protect molecules from radiation damage. But an interesting result was found in case of certain cancer cells. Here, dinoxanthine work more like a pro-oxidant than an antioxidant. And there are many such studies. I'm just showing you glimpses of some of those studies. So over here, for example, 3 different cancer cell lines, the names are not important, is only to show you that with increasing concentrations of dinoxanthine added to them exogenously in rather low concentrations in micromolar levels, all 3 cell lines lose their viability. If you look at, and they do so because these cells undergo apoptosis. So they also looked at the apoptotic cells and found that in all 3 cell types compared to non-apoptotic cells, the number of apoptotic cells was phenomenally higher. Apoptosis was, of course, looked at in terms of chromosome condensation and fragmentation. And all these red arrows indicate such situations in these 3 cell types, which are without dinoxanthine and with dinoxanthine. And 3 important results which support the apoptotic role of dinoxanthine in these cancer cell lines and more show the following. In every single case, addition of dinoxanthine increased ROS inside the cells. So it's a pro-oxidant role. Incidentally, many molecules work as pro-oxidant or antioxidant depending on the partial pressures of oxygen present there and the microenvironment inside the cell. So this is not a surprise that the same molecule which is known as antioxidant can also work as pro-oxidant. 3 other hallmarks of apoptosis were also detected in this. I am not showing you those results, but you can refer to this paper and it will show you all these details. Procaspases were degraded, BCL2 expression was decreased, and BAX expression was increased. All 3 are hallmarks of apoptosis, which clearly went to say that use of this molecule, an orange pigment from Dinococcus, resulted in apoptosis and regression of cancers in certain human cancer cell lines. It has also been tested in animal model systems, but I will not go into that detail, just to show you another non-nuclear application of one of the molecules, dinoxanthine. In fact, dinoxanthine is a very potent molecule, and the research on Dinococcus has gone now out of this world. For example, over here is a case where, very impressed by Dinococcus' phenomenal abilities, people have also tested if it can survive the harsh environments of Mars. In fact, when the first landing on Mars took place, pictures of rocks were published on which were found some single cells. And I was questioned in one of the highest bodies in BRC to ask if this could be Dinococcus. The answer, of course, was no. But encouraged by that feeling, lot of people in NASA and outside, retired scientists of NASA now outside, have tested such possibilities. In this case, it's a different Dinococcus strain, Geothermalis, which occurs in hot springs, is radiation resistant as well as thermoresistant. And they tested this under a whole variety of conditions, which are some of them are listed here. Prolonged desiccation. Bacteria normally do not survive more than 3-4 days of desiccation, but Dinococcus can survive more than a year or two of desiccation. Extremes of temperatures going up to 65 degrees and -20. Vacuum, which is typical of space, and simulated Martian atmosphere. Don't ask me how they created it. That paper here gives full details of that. And UV, which is very, very highly prevalent in space. And they also created combinations of these stresses. For example, to simulate space, they created combinations of desiccation, vacuum, and UV. And to create Martian atmosphere, they created combinations of desiccation, Mars atmosphere, and UV. And it's a long paper, as you can see, but the summary states these statements. First is that the substantial proportion of bacterial population, that is Dinococcus, remained viable under all stress conditions tested, including space or Martian atmospheres. In most cases, the biofilm form proved more advantageous for surviving space and Mars-like conditions. Now, over here are two pictures of biofilm form and the planktonic form of Dinococcus cells. They have been stained with either cyto9, which gives the green color in viable cells with intact membranes, and propidium iodide, which stains the dead colors or membrane permeabilized cells red. And as you can see, compared to the planktonic forms, the biofilm forms show much more resistance. But the more important conclusion is in the next slide, that leave aside the fact that Dinococcus survived even in situations, particularly under ultraviolet radiation, the cells lost their culturability, but they retained many of their components, in particular dinoxanthine, which was found to be quite stable and active even after prolonged exposure to space and Martian conditions. Dinoxanthine is now being, I'm told, being used by NASA or other space agencies to test the possibilities of testing life forms on other planets, because if dinoxanthine survives those conditions, there is hope that other life forms may survive. So under such conditions, the conclusion was that prolonged UV exposure with other combinations made the cells sometimes non-viable, but retained them in a viable but non-culturable, the BBNC state, as microbiologists know very well. And in particular, the pigment dinoxanthine was protected and active even at that stage. But what else can you do with Dinococcus? It's very interesting that Dinococcus has two surface layer proteins. One of them is called HPI layer. The HPI layer you can isolate from Dinococcus cells. It's the outermost protein of Dinococcus, and it comes out like a transparent pellicle. It's very easy to isolate HPI. We have done it many times. And the most interesting thing about it, it is a self-assembling nanomaterial. So it assembles like a layer, and this is the detailed structure of this layer. And people found it very interesting to deposit nanoparticles on it. So over here is the same HPI surface layer on which gold nanoparticles have been deposited. So nanoparticles, as you know, is a growing field. They are being used for all kinds of applications. I'll show you one or two in the next slides. But to be able to use these particles not as particles, but in the form of a layer or a membrane or a substrate, HPI layer seems to be a very good choice. And a lot of nanomaterials are now being deposited on HPI layer, because it's a self-assembling nano layer. We, of course, used HPI for a very different reason. HPI being the outermost layer, we thought we could use it for surface display of useful proteins. Now, why do you want to display proteins on the surface? You want to, for example, display antigens on the surface so that you can raise antibodies. We also want to deposit molecules which are active in bioremediation. For example, over here, we used HPI layer to display two kinds of molecules, the acid phosphatase as well as a metallothionein gene, which sequesters heavy metals like cadmium. And this paper was published only this year to show you that such surface displayed metallothionein and acid phosphatase could sequester and precipitate uranium and cadmium much better than Dinococcus cells, which also do this job, but these are able to do much better because here the molecules which will remove heavy metals are displayed right on the surface. Now, nanoelectronics is a big field today, and people try to grow nanowires for microelectronic devices, miniaturized devices, etc. And over here is one such result where again HPI layer of Dinococcus was used to deposit germanium. Germanium can be grown into wires. Normally these wires go haywire, but when deposited on surface layers like these, they grow vertically needle like these. And over here is a picture of 20 millimeter size nanowires grown on biomaterials. These are called biotemplated silver nanoparticles on which they grow germanium wires or even gold nanoparticles. People have also tried to make noble metal nanoparticles, particularly silver and gold are very popular for two reasons. Silver nanoparticles have a strong antimicrobial activity. Many of the microbiologists in the audience would know that. Gold nanoparticles have catalytic abilities of various kinds. They are particularly used in reducing several dyes, for example. And when very interesting study was published a few years ago, 3-4 years ago, where they found a very simple method to prepare silver or gold nanoparticles or even bimetallic nanoparticles containing both silver and gold, which is not an easy thing to do, I'm told. And the approach was very simple. They took Dinococcus cells with, of course, the HPI layer. Then they stressed it, and the stress was applied in the form of high concentrations of either silver nitrate or gold chloride. So the materials which you want to deposit as nanomaterial itself was used as stress, either silver or gold. And then they removed Dinococcus cells from it. So now there is only supernatant, and supernatant of Dinococcus is a wonderful preparation, as I will tell you in the last part of my talk. So they removed Dinococcus cells. They took this culture filtrate, which has been stressed with silver or gold, and now subjected it to the particular material of which they wanted to make nanoparticles. So when they stressed it with silver here, they could deposit gold nanoparticles very well. When they stressed with gold, they could deposit silver nanoparticles very well. And these are pictures of that. So this here silver was used as a metal stressor, and the red part is silver particles, the green part is gold. You can see the amount of gold nanoparticles deposited. Over here, gold was used as the stressor, and the red part is gold, and green part is silver. You can see the amount of silver deposited there. And then they also tested such noble metal nanoparticles for the desired activities such as antimicrobial activity. They tested it on a gram-negative organism like E. coli and a gram-positive organism like Dinococcus itself. Both worked very well. And they also tested gold nanoparticles for dye reduction type of activities and found that with increasing time, it reduced more and more dye. Now, how much of this is a real application or will become a real application, future will tell. But it's only to tell you interesting possibilities that Dinococcus holds for fields other than nuclear technologies, be it treatment of cancers using some kind of chemotherapy, be it in surface display of molecules such as antigens or others, or be it making of nanowires and nanoparticles. I will end my talk with this last story of Dinococcus, which is very interesting and is very exciting about Dinococcus at the moment. And this will take about 5-6 minutes. I mentioned about Dinococcus culture filtrate. Not only the cells are very interesting, the culture medium in which you have grown Dinococcus cells, the spent medium or the culture filtrate is even more interesting to some people. For example, Daly's group, this paper appeared almost 10 years ago and was the first paper of the kind. A massive work in this paper. Some of you are interested, you can look at it. They prepared culture filtrates of all kinds of molecules. So this is a Pseudomonas species, E. coli, Thermophilus, Dinococcus radioduscens, and this was a control. So they prepared culture filtrates of these organisms. Why? They wanted to test if this culture filtrate had any abilities to sequester ROS, because their a priori assumption was that since Dinococcus survives such high level of radiation, it will be possible to do so using other things than what we know of. What they found is that Dinococcus culture filtrate did not oxidize proteins post-irradiation. In all other culture filtrate, proteins were oxidized very badly. This is measuring protein carbonylation using antibodies. In case of proteins which were treated with Dinococcus culture filtrate, there was no protein oxidation. In another manner they tested, one of the very radiation sensitive restriction enzyme is BAMH1. All of you may have known or used it. And this, when irradiated, loses its activity. Now, when this BAMH1 was incubated with culture filtrates of all these organisms, in no other organism the activity could be protected after radiation. But in Dinococcus, up to a good amount of radiation, 4 or 6 kilogray, the BAMH1 activity was protected. Not only this, they now stored BAMH1 under strong desiccation conditions up to 66 days, and then tested the ability, stored it in desiccation in presence of culture filtrates of these organisms. And what they found was that at 0 day, all of them retained activity, but not after several days. Only Dinococcus retained that activity, particularly after 66 days, only Dinococcus culture filtrate had that ability. In short, Dinococcus culture filtrate has something that protects molecules, especially proteins, from strong. Excuse me, can somebody mute themselves? I have just 5 more minutes to take. Please do not sing.
Yeah, just a minute. Sir, I guess you have got muted. I'm just trying to unmute you. Apte, sir. Sir, can you unmute yourself?
Am I audible now?
Yes. Thank you, sir.
All right. So I will get back to just a couple of more slides are left. So finally, they came down to this preparation. This is what was present in culture filtrate and caused the protection. This is 25 millimolar potassium phosphate buffer, 3 millimolar of a decapeptide. I'll tell you what that decapeptide is, and 1 millimolar of manganese chloride. All three, if you add together, the solution has tremendous ability to protect the protein and its activity, including one of the most radiation sensitive enzyme, BAMH1. And what is this decapeptide? They analyzed the free amino acids present in the culture filtrate and found that these 10 amino acids shown here were most abundant and made a synthetic decapeptide out of it without bothering about the sequence in which these amino acids occur. This sequence was found to be the best. And this preparation, they now call it MN-DP-PI, MN for manganese, DP for decapeptide, and PI for phosphate. So now you don't need Dinococcus anymore. You can add 25 millimolar phosphate buffer, 3 millimolar of this decapeptide, and 1 millimolar manganese chloride, and you have a solution that can protect your proteins tremendously from radiation damage of doses as high as up to 20 kilogray, which normally nobody exposes anything to. And so what is the use of this? That's what is shown in my last slide. This is for making, now being put to use or thought of being put to use for making vaccines. The first experiments were done with bacteriophage lambda. The idea was to see that if they incubate lambda in this kind of extracts, and irradiate it with unbelievably high doses, what happens to it? What they found was that higher doses of lambda, higher doses of radiation killed lambda even in presence of MNDP-PI, destroying its DNA. After 20 kilograms, no DNA was left there, but the head and tail proteins of lambda were intact even at that dose, and these were also detected using specific antibodies to them. So what it suggested to them are the following. First of all, sterilization of pathogens with gamma rays is an attractive approach for development of inactivated whole organism vaccines, because you retain the proteins or the antigenic epitopes of these proteins even after such level of radiation. while completely killing the organism and destroying its DNA. But at such high doses, normally radiation doses required to ensure sterility also destroy immunogenic epitopes needed to mount a protective immune response. But not so if you use this MNDPPI with this protein. So we report here genome damage and killing can be uncoupled from epitope damage using a reconstituted manganese peptide complex of Dinocockers radiodurans. And these people themselves went on to create a vaccine for Venezuelan equine encephalitis virus, which is a deadly virus which can be spread as an aerosol. It has been used as a bioweapon for bioterrorism also. And so far, there is no approved vaccine or antidote against this. And they showed that when this entire virus was treated at very high doses of radiation, it destroyed its DNA and pathogenicity using this cytopathic assay, but retained the epitopes which cross-reacted with the antibody that they generated. And they have created a vaccine for VEEV. Subsequently, similar vaccines have been created in subsequent years using the same approach of irradiating the whole organisms in presence of MNDPPI at very high radiation doses. And two such vaccines are, one is VEEV, other is chikungunya virus, and third one is poliovirus. So all three vaccines have been cleared in animal model systems and are now undergoing clinical trial apparently. And this is particularly relevant with reference to the COVID-19 pandemic at the moment. And I want to end my talk by just saying that, as you have seen, that most of the vaccines that we have today are more specifically against one or two proteins, in particular the spike protein. Some have been raised using protein, some have been raised using messenger RNA, but they are directed against one or two specific proteins. And the virus is very clever. Within transition from Delta to Omicron, it has thrown more than 30 mutations. And now it's beyond recognition of all existing vaccines. As you can see, fully vaccinated people are also now getting infected with Omicron. And in the light of that, a vaccine raised against, let's say, all the protein epitopes of the virus or against the whole virus would be very desirable. And of course, a deadly virus like SARS-CoV-2 COVID-19 cannot be used unless it's completely activated and degraded at the DNA level. And that's possible by irradiating it in MNDPPI at extremely high doses of radiation. And practically everybody who has produced vaccines using such approach is at the end of their paper is now only talking about this. So hopefully this will be used for producing newer vaccines, not only against COVID-19, but many other deadly organisms, both bacteria and viruses, by irradiating them at extremely high doses in MNDPPI. I think I will close this presentation at this stage by thanking all these large number of people. I hope I have not forgotten some names. These are all the people who have worked with me on various aspects of Dinocockers. Many of them are PhD students. Some of them are from Bombay University. Many of them are my colleagues in BRC or in other universities, especially Pune and Baroda University, etc. Including some students who have been my eyes and ears and have read a lot of material for me in CBS as well, who are part of this list. Thanks to all those who gave us various strains and vectors to do some of the work that I presented. And I'm grateful to all those people whose work I have been able to cite today. And finally, a big thank you to all of you for your patience and for your patient hearing. Thank you. I'm happy to take any comments, queries, questions now. Thank you.
Thank you, sir. The session was amazing and there is so much to explore. We really appreciate your efforts in presenting so many untold stories of Dinococcus radiodurans for the students on a platter with your clear interpretation and getting it to the level of our UG students as well. The students will surely explore more in this field. I also welcome our principal, Dr. Anita Kanwar, ma'am, who has joined the session. Good morning, ma'am. Ma'am will address the audience after we take up the questions. Please be with us, madam. The chat box has been teeming with questions. I now request Dr. Dona Joseph, ma'am, and Dr. Malay Shah, sir, to take up the questions on their behalf. Over to you, ma'am and sir.
Uh, Good morning to everyone. Thank you, sir, for that very interesting talk on Dinococcus radiodurans. As Shweta ma'am said, there have been a lot of questions, and we'll start with a simple one. It's a doubt that I have. How did this organism evolve? What are the current insights on its evolution?
Okay, normal. Sorry, can you hear me?
Yes, sir.
Yeah. Normally, by normal principles of evolution, one believes that when an organism gets stressed continuously with a particular kind of stress, it slowly adapts to it, learns to live with it, and eventually that becomes a trait of that particular organism. Unfortunately, or fortunately, such high level of radiation has never existed on this planet. Even at the time of Big Bang, when the Earth was formed, the radiation levels were pretty high compared to what they are today, but nothing comparable to what this organism can tolerate. And the answer that is available as to how this organism gained such phenomenal abilities is kind of indirect. And the answer is that the organism did not learn to do all this radio resistance and DNA repair in response to high levels of radiations because they never existed. It learned to do this in response to extreme prolonged desiccation. And the reason for believing this are the following. Desiccation, especially prolonged desiccation, produces same kind of DNA damage that chronic exposure to or acute exposure to radiation does. Radiation will do it in a few minutes of time. Desiccation will do it in few hours or days of time, but both produce identical kind of damage. Also, Dinococcus is extremely desiccation tolerant. Among bacteria, it holds a record. It has been known to survive even beyond 2 years under completely desiccated condition. The other reason for this is that Daly's group and even Batista's group earlier isolated chemically induced mutants of Dinococcus, all of which were radiation sensitive. What they also discovered, these are close to 106 such mutants. What they also discovered that all these radiation sensitive mutants were also desiccation sensitive, which is not what they isolated them for. They isolated them for radiation sensitivity, but almost each and every one of them was also found to be desiccation sensitivity. And last kind of evidence is that if you subject Dinococcus to prolonged desiccation or radiation, and then look at the proteomic changes over next few hours or days, you get similar proteins going down and similar proteins going up in response to both of these two otherwise totally unrelated stresses. Now, these are not, I would say these are not unequivocal evidences that this is what it means. But these are fairly indicative that what Dinococcus can do with radiation, it certainly is able to do with desiccation, and most probably it learned to do this with radiation because it knew how to deal with prolonged desiccation.
Thank you, sir. That's really interesting to know. Also, this organism is resistant to acid, cold, and many other extreme conditions. Could it be that the same genes are responsible for all of these properties?
No, not at all. In fact, you will be surprised to know for the phenomenal stress tolerances this organism has. For example, apart from radiation, UV, practically every genotoxic stress that you can think of, even mitomycin C or hydrogen peroxide or whatever, it tolerates phenomenally high levels of these. Even simple stresses like nutrient starvation, you can put this organism, wash it thoroughly from a rich medium, put it in distilled water, put the tube in fridge for 2 years. After 2 years, take it out, let it out, you will still get 80-85% survival. Complete nutrient starvation. Desiccation also. So while it is so extremely tolerant to almost extremes of many other stresses, it is surprisingly very sensitive to two very commonly occurring stresses such as salinity and temperature. Very interesting. Optimum temperature for most bacteria such as E. coli is 37 degrees. For Dinococcus, 37 degree is lethal. It grows optimally at 32 degrees and not 37 degrees. E. coli, which we grow in luria broth, which has sodium chloride in it, if you will recall, Luria broth cannot be used for growing Dinococcus. We cannot have salt there. We grow it in so-called TGY medium, tryptone, glucose, yeast extract, no salt at all. So organism which survives otherwise very extreme stresses is very sensitive to these kinds of stresses. It is sensitive to many heavy metals, uranium, etc., but can be engineered to become resistant to them and remove them. The most important feature is radiation resistance, which is what is being made use of. And over and above that, you can add many things. The converse of this question is also answer is no, you cannot take 1, 2, 5, 6, or even up to 10 different genes from Dinococcus and put it in another organism and make that organism radiation resistant. That's why the question of making a acidophile or alkalophile radio resistant just does not arise. It cannot do. Easier is to make Dinococcus tolerate acid, because that depends on a few genes which you can add to Dinococcus, but not other way around. So the genes for the different stress tolerances are different. What is common to Dinococcus radiation resistance is only desiccation resistance and no other resistance.
Thank you, sir. I call upon Dr. Malay Shah to take the next question.
Yeah. Good morning, sir, and good morning, everyone. The next question is asked by A.R. Narayan. Can we find Dinococcus in our surrounding? If so, what samples we should use for the isolation? And there is a parallel question which has been asked by Payal Jaiswal. What are the things which are required to cultivate this Dinococcus?
Okay. The first question as to where all Dinococcus is found, meaning what is its common habit and habitat, let's say. It has been found practically everywhere. In fact, looking at it from same BRNS, which is funding this particular event, we got funding and did a project with 5-6 different institutions in the country, including Baroda University, to try and isolate this organism from all different kinds of habitats. And believe me, it could be isolated from UV exposed walls of Konark Temple. It could be isolated from Run of Kutch. It could be isolated from air in any room. It could be isolated from a very strange situation from pigtail hair as well. That's also recorded in the literature. So there is no specific ecological niche that this organism occupies. We have isolated it from just the air in the lab. Just take a Petri plate, move it in the air, open the lid, move it in the air, and if you are lucky, it will grow on that. The way to enrich it is one of the simplest. Whatever you have collected, just irradiate it and then plate it out. Only and only Dinococcus, if it is there, will grow very easily. So again, I want to repeat that while it is radio resistant, it's not a radiophile. It doesn't like to grow in radiation. It just dominates high radiation environments because at that radiation, everything else gets knocked out. Sterilized operation theaters, sterilized medical equipment, all have Dinococcus on them at many times. Only because everything else is dead and it has the entire field to itself in terms of space, in terms of nutrients, etc., etc. So it's, in short, from where to isolate it, you can isolate it practically from anywhere. Normally, it's present in very low abundance. So to enrich it, whatever you have collected, try to irradiate it and then plate out. It will eliminate the competition and Dinococcus will grow. Does that answer both the questions?
Yes, sir. Thank you, sir. Over to you, Dona ma'am.
I can't hear you.
Dona ma'am, you are on mute.
Oh, sorry. There is a question by V.B. Tilak. Are there any studies whether this bacterium played a major role in the restoration of greenery around Chernobyl?
I don't know of any such study which will show that this organism played a role in restoration of greenery. A priori, there is no reason to believe that. It's a heterotroph. On its own, it cannot grow anywhere. It needs some nutrition. When I say it's nutrient starvation tolerant and desiccation tolerant, it will merely survive there, but it will not grow luxuriously unless you give it a medium. Greenery, I mean, you refer to autotrophs, which will take over, and they have to be specifically planted there. So no, I am not aware of any study where this organism played a role. The question could have been whether in the initial days after Chernobyl exposure, whether this organism dominated there. Unfortunately, and I call it unfortunate because one would have very much wanted to see if this organism dominated the Hiroshima exposure sites or Chernobyl exposure sites. There are no such data available. But it dominates, for example, in US, there are several places where during Cold War, nuclear stockpiles were stored for 40 years, 50 years, and they leaked. Their casings, the metal casings got corroded, they leaked, turning the lakes radioactive. Some of these are very famously located in important nuclear institutes in USA. And in all such situations, Dinococcus was found. So it does dominate high radiation environment, but as to Chernobyl or Hiroshima or Fukushima for that matter, there are no studies to see if that organism dominated or whether it had any role at all in restoration of greenery subsequently.
Thank you, sir. Over to you, Malesa.
Sir, there is a question asked by Ratnakar, sir. How does 200 base pair break after gamma dose reassemble back and become functional without misalignment and making mutants rather than a functional molecule?
First of all, thanks Ratnakar for seeing you after a very long time. I don't know why you asked this question for 200 base pair. Basically, the dinococcal genome, when you irradiate like this, gets close to 1200 to 1400 double strand breaks after about 6 kilograms of radiation. And believe me, other bacteria like E. coli or Bacillus cannot stand even 2 double strand breaks per genome. This one survives that many. And at that stage, the DNA is fragmented, or average fragment size is less than 0.5 kilobases. So the way it repairs itself is, first thing that it does is it has a unique DNA repair phenomenon, which was published in Nature several years ago. It's called ESDSA, for Extended Synthesis DNA Strand Annealing, which means that whatever are the fragments created, blunt ends or staggered ends, it just adds some more bases of single-stranded DNA to it randomly, totally randomly, not in a templated manner, and then uses non-homologous end joining initially to stitch together all pieces totally randomly. These are not homologously recombined pieces, but the idea is to save loss of any DNA because any staggered ends will be chewed up by exonucleases of all kinds. So very first thing that it does is that it stitches back its chromosome totally incorrectly initially in the first few hours of post-iteration recovery. And subsequently, then it applies its recombination pathway, which is highly proficient, including REC-A and FORJ, to find homologies and put back things in the correct places to regenerate its entire chromosome without any single mutation. So that's what is important. And this initial part of extended strand DNA synthesis and annealing and non-homologous end joining is what seems to be responsible for its tremendous recovery. Beyond that, it has multiple copies of genome, as I told you. And very surprisingly, these multiple copies are held together by means of DNA binding proteins. In fact, if you look at the picture, the DNA occurs inside this organism in the form of a toroid ring around the periphery. And even after irradiation, the toroid ring is seen, meaning though the DNA is fragmented, the fragments don't diffuse out, they are available nearby. And although the DNA gets damaged, some good copy of every gene probably is found, is replicated and ligated and repaired. It's only when the dose exceeds so much that not even one intact copy of certain genes is available, then it starts showing sensitivity, and that's the reason why it cannot withstand beyond 15 or 30 gray. But otherwise, that's how it repairs itself.
Thank you, Dr. Apte.
Yeah, welcome. But I should also say that there are many facets of DNA repair in this organism which are still unknown. And there is ample scope to study DNA repair further. Fortunately or unfortunately, first 10, 20 years saw nothing but DNA repair studies on this organism. And then people got, I suppose, a little fed up and diverted to other areas. But DNA repair still remains sort of incompletely understood in this organism and can be explored and should be explored further.
Yeah.
Yes, thank you, sir. There is a question by Abhijit Shankar and a related question by Disha Biswas. The first question is, can we engineer the radiation resistance genes into other bacteria and remove the DNA repair genes like SBCSC, SC, sorry. And Disha Biswas asks that if we genetically engineer other bacteria to have PPRL genes, will they also help in apoptosis?
Okay, these are two different questions and both have very interesting answers. So I will answer them one by one. First is the idea that can we engineer another organism to become radiation resistant by putting one or many genes from Dinococcus and removing the genes that Dinococcus lacks. I hope whoever has asked this question, sorry, I forgot the name.
It is Abhijit,
yes.
Abhijit Shankar.
All right. So I hope you have tried to genetically engineer organisms. Even engineering one gene used to take a lot of time earlier. To engineer an organism with so many genes is going to take a lot of time, and it's sort of asking for the moon, actually. And even in Dinococcus, all these genes on their own do not do much. The whole context is required. It's not only the genes, the proteins, the metabolites, the whole cellular microenvironment is important for the kind of radio resistance you see in this organism. There are other organisms which are fairly radio resistant. For example, Bacillus subtilis spores are very, very radio resistant. They can survive up to 10-12 kilogram of radiation, but only when they are present as spores, which means when they are in the non-growing state. The moment they start growing, they are as sensitive as E. coli. So in short, good luck to you if you want to engineer any organism with so many genes and hope to make it into Dinococcus. But I wouldn't try any such thing. Number one. Second, what was the question? The second one, if I can repeat. Yes, about PPRI.
Yes.
About PPRI. Yes, that's a very interesting question. I didn't touch upon it, although I said earlier and even just now that taking genes from Dinococcus, putting in another organism to make it radio resistant may seem like a very far off idea because too many genes. But there is one gene of Dinococcus that has been put in many organisms, and that is PPRI, the protease which cleaves DDRO in Dinococcus to cause radiation-induced gene expression and is largely responsible for the post-irradiation repair and radioresistance of Dinococcus. For no reason has been put into all kinds of organisms, including probably 24 of bacteria, plant cells in tissue culture, animal cell lines in tissue culture, and even animals like mice. Again, as I said, for no reason, only in the hope that because it makes Dinococcus radio resistant, can it do something in other organisms? And lo and behold, the answer is even shocking. In every single case of such gene transfer, PPRI gene transfer into other organism that has been reported and published, it has had beneficial effects. Every single bacterium, E. coli, Pseudomonas, Lactobacillus, you name it, every single organism now becomes more stress tolerant. Sometimes they may become salt tolerant, sometimes they may become desiccation tolerant, sometimes they produce more ethanol, sometimes they produce more lactic acid, and so on and so forth, for reasons which we don't understand at all. And this becomes even more puzzling when you know what PPRI is. PPRI is a gene which among all the known genome sequences is present only in Dinococcus so far. Number one, its target, the known target, the DDRO gene and protein among all the genome sequenced is found only in Dinococcus, nowhere else. So there is no reason why this protein should do anything in any other organism, but it does. And a lot of people are trying to find out how and why it does. But the answer is yes, PPRI put in other organisms has had some magical effects. And I would feel, or one would feel more confident to do this further if one knows how PPRI does it, rather than simply trying it as a blind shot and hoping for the best.
Thank you, sir.
Sir, the next question is asked by Hussein. Do we know why dinoxanthin acts as a pro-oxidant specifically in cancer cells? And there is another question which has been asked by Neha Sawant on similar lines. Under which microenvironment conditions does dinoxanthine act as an antioxidant?
Okay. Dinoxanthine in vitro in solutions, for example, has whenever been applied exogenously to see if irradiated proteins or other molecules will be protected. It has always worked as a raw scavenger and antioxidant. Why inside the cancer cells it works sometimes as pro-oxidant, not always though, but sometimes work as pro-oxidant is not very clear. The experts say that practically every molecule which can work as antioxidant can also work as pro-oxidant depending on its own concentration, depending on the partial pressure of oxygen available at that time, and other things from the cellular microenvironment. So take it that the answer why an antioxidant turns into a pro-oxidant sometimes is not very well known, but in certain situations it happens to not only dinoxanthine, many other antioxidants also turn out to be pro-oxidants. Many studies on cancer cell lines have shown that, and that is true here. So at least the paper I showed you is one where 3 different cancer cell lines were tried, but literature is full of at least half a dozen papers. I saw half a dozen of them yesterday itself, or more maybe, where dinoxanthine has been found very effective versus many other proteins which have been tried, such as lycopene or other xanthophylls, or compounds like plumbagin and all kinds of natural plant products which people have tried to use as antioxidants.
Thank you, sir. One last question, an interesting question from Ratnakar, sir. Okay. In pharma, geobacillus is used as a biological indicator for sterility validations in autoclaves. Can we use Dinococcus radiodurans as a biological indicator for radiation dose validation?
Sure, you can use Dinococcus in that capacity. At the moment, what is being used, because usually more than 12 log cycle loss of viability is taken as a sign of sterility. At the moment, what is being used are Bacillus subtilis dried spores. They are sort of stuck to all medical products which are sterilized in BRC, for example. And as long as there is they are used as the internal standard. But sure, you can use Dinococcus, but it has not been put to use of this kind so far. In principle, there is no reason why it cannot be used.
Thank you.
Sir.
Welcome.
Sir, I will take one more question, last question. This was asked by Susan ma'am and also from Shantini Nair ma'am. For radiation sterilized product, is Dinococcus radiodurans a problem?
Microbiologically speaking, it is a problem, because when you want to sterilize, you don't want to see any living organism around, and this would be around. However, it's not a problem of the clinical kind, because fortunately, and most fortunately, it is a non-pathogen. It doesn't affect humans, or for that matter, any other animal or bacterium or fungus to cause a disease. In fact, when it was first discovered, it was just after the Second World War, when American soldiers were given irradiated meat cans to eat when they were working in Europe. It was in one of those cans, when it was opened, an orange scum was found at the top, which was all Dinococcus. That's where it was discovered. And that caused a great concern, because if it was a food pathogen and pathogenic to humans, it would create disaster, because many irradiated cans would have that. But that was not the case. So to answer your question, it will not give you perfect sterility as a microbiologist would desire, but it will still not be a problem in the sense because it's a non-pathogen. You can live with that level of Dinococcus. Fortunately, it's not a very abundant or luxuriantly growing Dinococcus under those situations, because in sterilized medical products or surgical equipment, etc., no nutrients are available. But if you just want to see if anything is there at all, yes, Dinococcus could be there at times.
Thank you, sir.
Welcome.
Thank you, sir, for that very interesting, the interesting answers to all the questions. There are many more questions, but I think we don't have the time now to take them up. So I hand over the mic to Shweta.
Can I make a suggestion?
Yes, sir.
But, it has usually happened that my talks are shorter and question answer sessions are longer as far as Dinocockers talks are concerned. So that's why I mentioned my email there
Yes.
that people could use to send me these questions afterwards. And I'll be happy to answer as many of them as I can. Please don't ask tough questions, ask easy ones so that I can answer. Thank you.
Thank you, sir.
Thank you, sir, for patiently answering all our questions. And we will surely be giving your email ID to the students so that we get all the answers to them. And we will be in touch with you for more study into this bacterium. I now request our principal ma'am to address the gathering. Over to you, madam.
Thank you, Shweta, and good morning, everyone. First of all, I would like to apologize for being late because I had some other commitment. And our esteemed guest and speaker for the webinar, Professor S.K. Apte, President of IWSA, Dr. Lalita Dhareshwar, Susan Madam, and other dignitaries from IWSA, our Vice Principal and Head of the Microbiology Department, Dr. Shantini Nair, former heads, staff members of Microbiology Department, all invitees, and my dear students. Thank you all for joining this wonderful session on behalf of Vivekanand Education Society's College of Arts, Science and Commerce, which was organized in collaboration with IWSA. To give you a brief introduction of our college is multi-faculty college, 42 years old, and we are permanently affiliated to University of Mumbai. And currently our college is running 29 courses in aided and self-financing section, which includes 17 undergraduate programs, 9 postgraduate programs, and 3 PhD courses. We have received A grade from NAAC, that is National Accreditation and Assessment Council, into consecutive cycles, and we have also received Best College Award from University of Mumbai under urban category. Recently, we are awarded with autonomous status by UGC, and which is also confirmed by University of Mumbai. So all this became possible because of very hardworking and dedicated staff members and excellent exposure which is given to our students by having continuous collaborations with international and national level agencies and institute of repute like IWSA, Center for Basic Sciences, TIS, TIFR, BRC, HBCSC. We have regularly organized programs in collaboration with the institutes of repute, and I take opportunity to thank IWSA for giving us chance to organize this popular lecture by an excellent speaker, Dr. Professor Apte. Sir, thank you very much for wonderful talk. Though I joined late, but whatever I have grasped was very interesting, and the topic was so interesting that it will definitely motivate some of the young researchers in our college, and they will contact you. Sir, we would appreciate if you could take out your time and help these researchers to do research in such a interesting field. Thank you so much, sir.
Thank you.
Thank you, Anita ma'am, for joining in and telling everyone about our college. May I now request the event coordinator for today's session, Suman Gangar sir, to propose the vote of thanks. Over to you, sir.
Thank you, Dr. Shweta Patil, madam. So it's my pleasure to propose this vote of thanks towards today's webinar. So at the outset, sir, I am very much indebted to you for accepting the invitation and sharing with us this research and your expertise on this Dinocockers radiodurans. It was a power-packed, neat and a wholesome presentation, sir. Truly a guide into the birth and the life of this strange and amazing organism and what we can do with it. Many people have thanked you today, and I don't want to bug more with all the thanks, but I really loved it. The way, it's amazing that this organism, which is so much ubiquitous, present everywhere, At one end it is so much resistant, and at another end it is not able to tolerate the salinity and all. So that clearly tells us that there are many faces to things which we learn and which we expertise on. Thank you so much, sir. It was amazing. Department of Microbiology and VESASC is a proud collaborator with Indian Women Scientists Association, IWSA. Thank you, Dr. Rita Mukhopadhyay, President IWSA, Dr. Lalita Darishwar Madam, Susan Yepan Madam, Sureka Zingre Madam, former presidents of IWSA, and other esteemed office bearers. I'm truly amazed to see their enthusiasm in taking science to students and the community at large, and I hope to imbibe some of your qualities. I thank all the department members of the microbiology in hosting this popular science lecture. A lot of coordination goes behind the camera, which results into this smooth transition for the audience. Thank you, Dr. Shweta Patil Madam, for comparing the event, Dr. Dona Joseph and Dr. Malesha for picking up the questions and moderating them. Special thanks to Mr. Shashank Joshi for giving us this technical support. Finally, I thank the head of the department and vice principal and the convener of this webinar, Dr. Shantini Madam, for inspiring us to always work towards such activities, giving us the freedom to construct the same and believing in us. Principal Madam, thank you so much for taking out the time and being with us, and the management for their kind support in all these endeavors. Thank you so much, participants, for being with us. I was so happy to see the numbers getting to 100 and then 200 and then it almost touched 250. That clearly shows that sir is well known as a speaker. At the same time, the topic is amazing. So thank you so much. I hope you are filling up the feedback forms and the certificates are reaching it to you. And I can see our alumnus, Dr. Subhojit Singh, raising his hand. Sir, please join in, please. Dr. Singh. I think I saw Dr. Singh raising a hand. So I guess maybe it was by mistake. I don't know. But I'm sure he is also part of CBS.
Yeah.
Yeah. Okay, fine.
Here.
Yeah. So that's I end my vote of thanks. Thank you so much.
Yeah. Thank you, Suman sir. There were many people who had joined into this session. We also have Jasantha ma'am here, Subhojit sir was there, and also Apte sir's student Dipti was there some time back. So I just thought I should mention, and along with that we have our Apte sir, Paul madam was there, and Hema madam was also there, and also teachers from the chemistry department were there. So I thought I should just mention that to you. So thank you everyone. The feedback link, I hope you all have filled it up. Your e-certificate will be sent to you in a week's time. Can I please request Suman sir to put up the feedback link once again? There are participants asking for it again. So as we have reached the end of this knowledge-packed session, I'm sure all will explore more of this much to be explored bacterium. Let us end the session with a quote of Sri Ramakrishna Paramahansa. As long as I live, so long do I learn. Let's stay strong, stay safe, since the danger of this pandemic is yet to stop. Thank you all for joining the session. Thank you.
Thank you. Thank you all.
Thank you, Dr. Apte.
Thank you.
Thank you, Dr. Apte.
Thank you, Susan.
Viveka, Anida Kanwar, Shantini, Suman, Donna, Shweta, Malaysia, and all the IWSA members and all the participants once again. Thank you.
Thank you so much, sir. We are truly indebted to you.
Thank you, sir.
Thank you. It would have been my pleasure to come physically to Vivekanand College or IWSA Vashi, but thanks to pandemic that was not possible. Let's look forward
we'll definitely look forward to
action in future. Yeah, I hope the pandemic goes away.
But sir, we were able to reach people in India and abroad. So that was something which we are happy. A lot of our students, alumni who are abroad, they were happy to have this session, sir. Thank you so much.
Yes.
Okay. Thank you.
Thank you, sir. We will definitely look out for an opportunity to call you physically. Thank you, sir.
Okay. Thank you. And I take your leave. Yeah.
Bye, ma'am.
Bye, ma'am. Bye,
Susan, ma'am. Bye, Sureka.
Positives.
Yes, ma'am. Yes, Sureka, ma'am. Thank you, ma'am.
Thank you.
Thank you so very much for making this happen.
Yeah, thank you.
Thank you all.
Suman, you have to put off the recording.
Yes, I will do. Thank you, Malay. Thank you. Bye, everybody.
Bye.